So on behalf of Oluwaseun supervisors, Jim, Saskia, and Michael and myself, I'd like to welcome you all to Oluwaseun Fatunla's completion seminar. And I acknowledge that today we're meeting on the unceded lands of the Wurundjeri people of the Kulin Nation and pay my respects to elders past and present. Oluwaseun trained in medicine and undertook a fellowship in anatomical pathology at University College Hospital in Ibadan in Nigeria. And in 2021, Oluwaseun received the competitive WEHI International IPSI Scholarship and came highly recommended following his outstanding scholarship interview. Jim and I had only just started the lab in late 2021 when we both received an email from Oluwaseun expressing interest in starting a PhD in the lab. And we couldn't believe our luck in securing both our first student and someone who had clinical training in pathology, a very highly sought after skill. So this was certainly an incredible opportunity for everyone, and we eagerly accepted Oluwaseun into the lab starting in late 2022. Initially, we had designed Oluwaseun's project to primarily harness computational techniques, leveraging digital pathology, AI tools, and analysis of methylation and sequencing data, pairing this with Oluwaseun's clean pathology eye, with Saskia as his primary supervisor. And as his first year went on, Oluwaseun diligently took R courses and tried to apply coding to the data that he had and was generating. But we ended up pivoting at his confirmation to focus on TMA generation and spatial proteomics to nurture his background, and then I had the joy of taking him on as the primary supervisor. Now, as you'll see today, Oluwaseun not only excelled in building his cohorts to interrogate the progression of low-grade glioma histologically, but also successfully completed advanced bioinformatic analyses of a complex methylation dataset and learned to analyse the spatial proteomics dataset in R with grace and great competency. So we are so proud of Oluwaseun and what he's been able to achieve during his PhD. So he ended up performing all the bioinformatics that we tried to take away from him and made findings that are highly novel and important to the field of glioma research, which we hope to publish with high impact in the next year. Oluwaseun has contributed broadly to the lab, in addition to supporting wider divisional projects, supporting in authorships in Nature Medicine, Neuro-Oncology Advances, a review article, and multiple other papers currently out for review. Oluwaseun received support from the Greg Simmons Philanthropic Top-Up Fellowship, and this year has not only had a baby, but has been back in the clinic, maintaining his pathology training with the goal to be clinically focused here in Australia once he very shortly and very successfully completes his PhD. Welcome to the stage.
Good afternoon, everyone. Today, I'll be presenting my PhD project, determining markers of poor progression and survival in low-grade glioma, supervised by Sarah, Jim, Saskia, and Michael. Thank you for being here, and I'm very grateful. So let me start with an incidence overview. Brain cancer occurs in only about 7 per 100,000 people in Australia. But what really stands out is that it carries an XX mortality burden, unlike breast, prostate, or some other cancer types, where survival has improved. Brain cancer still carries one of the highest mortality to incidence ratios in Australia and even globally. This gap highlights both the clinical and research challenge we are facing when it comes to brain cancers. This becomes even clearer when we look at brain cancer survival reality. Brain cancer kills more people under 40 than any other cancer in Australia. The 5-year survival remains poor at around 20%. And unlike many other cancers, we've seen minimal therapeutic progress over the past 3 decades. So despite having a relatively low incidence, its impact, particularly on younger adults, is profound. Gliomas are a major focus within the brain cancers family. They account for a quarter of all primary brain tumors, but more importantly, they make up about 75% of all primary malignant brain tumors. Gliomas in adults include astrocytomas, oligodendrogliomas, glioblastomas, and sometimes ependymomas, which is more common in children. These tumors are the main driver of poor survival outcomes in brain tumors. One of the major advances in understanding gliomas has come from recognizing the role of IDH mutations. Histology alone was previously used to define the glioma subtypes, but now the IDH mutation status is key and central. About 80% of gliomas are IDH wild type. These are the classical glioblastomas, while around 20% are IDH mutant, split between the astros and the oligos. This molecular stratification has reshaped both the classification and prognosis of glioma. The reason IDH is so important lies in its biology. Wild-type IDH fits normally into the TCA cycle, supports the alpha-ketoglutarate, and consequently, the dependent demethylation. In contrast, mutant IDH produces 2-hydroxyglutarate, 2-HG, which blocks the process and reshapes this epigenetic landscape. Clinically, this translates into a marked survival difference between patients with IDH mutant and IDH wild type, which are the more aggressive and treatment-resistant cohorts. This biology is now fully embedded in the latest WHO classification, which is the latest classification that was done in 2021. Diffuse glioma had first split by IDH status with ATRX and 1p19q, splitting them into IDH mutant tumors, which are further subdivided into oligos and astros. It is worthy to know that there is upscaling of astrocytoma grade 2 and grade 3 to grade 4 in the presence of the homologous deletion of CDKN2A/B. This represents a major paradigm shift in this latest classification. IDH wild type with necrosis and macrovascular proliferation, either alone or in addition to TET promoter mutation, EGFR gene amplification, or even gain of chromosome 7 or loss of chromosome 10 changes, are now defined as glioblastoma grade 4. From literature context, grade 2 and grade 3 gliomas are referred to as low-grade glioma, LGG hereafter, which is the main focus of my study, while grade 4 tumor constitute high-grade glioma known as HGG. These LGGs, as we have previously defined them, typically present in young adults, and they account for about 15 to 30% of all gliomas, with median survival ranging from 6 to 14 years, depending on the molecular features of the tumor. While their initial course is often more indolent than glioblastoma, they show highly variable behaviors with divergent treatment outcomes. Historically, treatment has been dominated by surgery, with further management comprising radiotherapy and even chemotherapy, which are usually guided by the age of the patient and the extent of tumor resection. Temozolomide, approved for glioblastoma some years ago, has since been investigated in lower-grade gliomas, with more recent trials supporting its role, and just last year, the FDA approved vorasidenib as being the first targeted IDH inhibitor that is used in gliomas, with some already in trials, clinical trials. This is a major milestone, but it also underscores how slow the progress has been, with only a handful of practice-changing trials in over 30 years. The patient trajectories illustrate why LGG remains such a challenge. For example, from this sketch, patient A was diagnosed at 24 years of age with grade 2 astrocytoma. Today, is alive 15 years later with stable disease. Patient B had a similar start, but progressed earlier and died within 10 years of diagnosis. Patient C, the disease transformed aggressively to grade 4 and died within 5 years of initial diagnosis. This heterogeneity, which is the patient following different courses, despite similar initial diagnosis, calls for better understanding of the tumor at molecular level. This leads to the categorization of the different outcomes of lower-grade glioma. Stable disease means tumor do not recur. Recurrence refers to tumor that come back at the same grade. For example, grade 2 coming back as grade 2. Progression, on the other hand, or malignant transformations, as some call it, refers to grade escalation, such as from grade 2 to grade 3 or to grade 4. Longitudinal studies show that by 10 years after diagnosis, up to 70% of LGGs will have undergone progression. Understanding what drives recurrence versus what drives progression is central to improving patient outcomes, and this is the focus of my PhD. Not all progressions are equal. On the left side of this slide, you see that malignant transformation from grade 2 to grade 3, or from 2 to 4, or a stepwise transition from 2 to 3 to 4 has a major impact on overall survival compared to patients who never progress. On the right, in addition to grade escalation, the timing also matters. Patients who progress early within the 5 years of diagnosis do far worse than those who progress later. This highlights that the need not just to talk about whether a tumor will progress or not, but when the tumor will progress. Despite this, we still lack clear predictors. Do current clinical pathologic factors really predict progression? Which molecular alterations actually drive progression? Can we define reliable biomarkers that guide therapy? And increasingly relevant, can histology-based AI models accurately forecast progression risk? These questions frames gaps that I will be addressing. Traditionally, we've relied on EORTC and RTOG clinical models, which include age, histology, the tumor size, and even the resection status. The status of the margin of resection has been the only consistent prognostic clinical pathological parameter. This remains useful, however, they don't fully capture biological behavior. Surgery may delay recurrence, but it does not stop progression. Tumor biologists drive that. As Perez study showed, a pure clinical prognostication is no longer sufficient to predict progression in glioblastoma. Noted earlier on, mutant IDH produces 2HG. This leads to widespread DNA and histone hypermethylation. This alters the immune microenvironment. Macrophages become immunosuppressive, reducing the MHC2, CD8T, and CD86. White T cells show reduced cytotoxic activity and increased PD-1 expression. Together, this creates a profound immunosuppressive tumor niche. As a result of this, IDH mutant LGGs are often described as cold tumors due to the low myeloid and T-cell infiltration, with reduced pro-inflammatory signaling. These tumors typically respond poorly to immunotherapies that rely on T-cell activities. The challenges ahead are clear. We do not have the full spatial mapping of T-cell infiltration, and we lack reliable TME biomarkers to predict progression or therapy responses. Now, moving on to AI application to glioma progression prediction, a study by Shirazi et al. Showed that routine H&E slides can be used with machine learning to define tumor microregions. These regions, with their associated level gene signatures, stratify tumor progression risk. By training on IVGAP, which is a publicly available data, where you can get a lot of materials on glioma and other brain cancers, then they applied this model onto TCGA GBMs, and they generated spatial gene programs that correlated well with survival and helped to predict progression risk. How did they do it? Here's the workflow. Spatially annotated RNA-seq data from IVGAP-defined regions was used to train the machine learnings. After segmentations and quality control, region-specific signatures were projected onto those TCGA samples, followed by enrichment and survivor analysis. This model by Shirazi et al. Achieved about 70% pixel-wise accuracy in predicting GBM prognosis from H&E slides. The Abigap studied key 5 histological regions in gliomas. The first one is the leading edge, abbreviated as LE. The next one is the infiltrating tumor, IT. Then we have the cellular tumor, CT, the pseudo-palisading necrosis, PAN, and the microvascular proliferation regions, which is the MVP. The leading edge reflects invasion program. That is where the tumor invades into the normal brain structures. The IT is linked to angiogenesis and immune infiltration and has been linked to poor survival in patients. The next one is the CT, which reflects the proliferation area of the tumor, and this is where we have the highest tumor density within the H&E. Next is the pseudo-palisading necrosis, which marks apoxia and stressed responses. And lastly, the microvascular proliferation is enriched for angiogenesis and also linked to poor survival. This region captures the core of biology of gliomas and form the basis for the AI prediction. We adapted this model that was used for GBM to our cohort, which is LGG. Bringing this together, the key challenges in LGG remains the unpredictable tumor behavior, the paradigm shifts in how we define risk, the clinical impact of early recurrence and rapid progression, the tumor heterogeneity and microenvironmental influence, and finally, the absence of reliable biomarker that we can use to predict stage tumor. These are the barriers we need to overcome if we are to improve prediction and therapy responses. This brings me to the aims of my PhD. My central hypothesis is that specific clinical pathological traits and spatial configuration of genetic changes influence survival and progression risk, and that artificial intelligence can be used or can be leveraged to predict clinical outcome or the risk. I structure my project around 4 aims. The first one is to define clinical outcomes in poor prognosis LGG. Secondly, to identify molecular drivers of rapid progression. Then next was to apply spatial analysis to recurrence and progression. And lastly, to adapt and test AI-based prediction models that was used on GBM to LGG. This project takes a multimodal approach. We integrate clinical registry data and sample recruitment, sequencing of DNA and RNA, generation of high-dimensional tissue imaging using MIBI, and of course, histology-based AI annotations. Together, we provide a complementary insights from patient-level outcomes to molecular drivers, to immune and macroenvironmental patterns, and of course, ultimately, computational prediction. So for the sake of this presentation today, I'll be focusing on the first 3 aims of my project. Patient details were identified through the brain tumor registry, and samples were recruited from the Royal Melbourne Hospital Anatomical Pathology Tissue Bank. Of the 732 patients that we identified with initial LGG tumors, 289 met the criteria for this project. Importantly, the pre-2021 classifications were updated to the recent classification using histology, immunohistochemistry, and sequencing data. Here is the classification process. Starting with the IDH1, tumors were stratified into mutant and wild type, with the initial wild type tumors excluded. Among the IDH tumors, ATXR, P53, and 1P19Q defined oligodendrogliomas versus astrocytomas. MTAP, which is methyl adenosine phosphorylase, is a surrogate marker for CDKN2A, which was used to upscale tumor where it is mutated from low grade to higher grade. Most of these samples were sent for NGS for molecular validation, allowing alignment with modern molecular criteria and fulfillment of the recent classification. When we reclassify this tumor using this recent classification, most diagnoses remained consistent with original reports. About 87% of these cases were unchanged, while only 12.5% shifted diagnosis. This shows that while most prior diagnoses were consistent, molecular reclassification was still very crucial. It identified the subset of cases that require updates, ensuring full alignment to latest criteria. Now, we noted that astrocytoma grade 2 shows frequent upscaling across surgeries in the red box there, including a later shift to higher grades, shown by the arrows. Here we note that most of the cases that actually shifted diagnosis were the astrocytoma grade 2. In contrast, oligos and astros grade 3 remain fairly stable in our diagnosis, in our reclassification system. When we stratify by initial grade, patients with grade 3 tumor did significantly worse than those with grade 2, as also noted and seen in several literature reviews. And when we stratify by tumor status, progression was associated with poorer survival compared to those that had pure recurrence. Both analyses underscore the importance of grade and tumor status as prognostic markers. Timing proves critical. Patients with early tumor events, even within the first few years of initial diagnosis, had markedly worse survival outcomes than those with later events. On the right, we see why therapy matters. Adjuvant treatment after initial therapy, significant prolonged time to recurrence or progression, extended the median interval from 37 to about 52 months. Here we maintain how different survival measures relate. Overall survival, OS, is mathematically the combination of progression-free survival and post-progression survival. Interestingly, PFS and PPS are inversely correlated because both form the OS. While PPS shows strongest correlation with OS, both of them contribute to the longer time overall survival, highlighting that survival depends on both the timing of progression and what happens after progression. Finally, this slide makes the contrast very clear. As earlier noted, the survival gap between recurrence and progression cohort is significant, raising a key research question. Can we identify molecular markers that tell us which patients will have pure recurrence and which patients will progress? To summarize this clinical arm of my study, astrocytoma grade 2 frequently upgrade to grade 3, unlike the more stable oligos. Both higher initial grade and tumor progression rather than recurrence are linked with worse survival. Timing also matters. Early event shortens survival, while adjuvant therapy delays recurrence or progression. And overall survival shows contribution from both the PPS and PFS, with PPS as the stronger determinant. We then turn to epigenetics. Methylation profiling, pioneered by DKFZ, is a German word, provides genomic-wide tumor classification beyond histology. It identifies subgroups with distinct tumor outcomes. As you can see from here, this is oligodendroglioma that was classified purely on methylation profiling, and this correlated well with the WHO diagnosis that we had in our cohorts. And not just on that alone, this DKFZ classifier has reclassified about 12% of CNS tumors across thousands of cases since the classifier was introduced. Today, methylation profiling is central to glioma diagnosis, risk stratification, and biomarker discovery, and it underpins our analysis of glioma evolution. From the 289 eligible EGG patients, we assembled a cohort of 111 matched pairs. This included 36 pairs with DNA extracted locally, plus 75 publicly available data sets from GLASS and TCGA. Together, this formed a reasonable, robust data set to interrogate longitudinal methylation dynamics in glioma. We applied the methylation classifier to our cohorts. On the left, you can see the mapping between WHO diagnosis and the methylation-based classification. Concordance was high, around 85% in our data set, similar to a previous report that reported a slightly lower value. This reinforces that methylation profiling is a reliable adjunct in glioma diagnosis. When we look at methylation longitudinally, we saw a significant overall decrease from initial to subsequent tumors. This suggests that global hypomethylation could be an important feature of glioma evolution. But when we splitted these cohorts, an important pattern emerged. Recurrence cases shows no significant change in methylation between surgeries, while in contrast, progression cases showed a marked drop, again, highly significant. In other words, hypomethylation is not just a feature of time, it is specifically associated with malignant transformation, meaning that what we saw at the global methylation level that we initially showed was purely driven by the progression cohort and not the recurrence cohorts. A smaller subset of patients underwent 3 resections, as noted earlier. This gave us a rare opportunity to track changes beyond the 2 time points and provided a valuable window into a stepwise process of progression. Here we see those dynamics. Recurrence remained relatively stable in methylation profiling, drifted between mild hypermethylation and mild hypomethylation. Progression, on the other hand, showed incremental hypomethylation across each time point with significant slope values. This progressive disease decrease in methylation underscores its role in driving tumor evolution. We then ask whether survival stratification could reveal aggressive disease gene. On the left, we saw stratified KM curves showed progression, early events, and even early deaths. Each identify poor outcome groups. On the right is the overlapping of these genes to yield a core set of 23 aggressive disease-associated genes linked consistently to rapid progression and early mortality. Here we look specifically at the 23 associated genes. Recurrent tumors tend to retain relatively higher methylation, whereas progression cohort shows broad hypomethylation. This reinforces the idea that widespread hypomethylation of aggressive disease gene is a molecular hallmark of true progression, distinguishing it from recurrence. These genes were validated using the TCGA RNA-seq data, which shows complementary expression changes. For example, genes that were hypomethylated were upregulated in the RNA-seq data, while genes that had a CPG hypomethylated were downregulated in the TCGA RNA-seq data, which is what's expected in literature. When we zoomed in on genomic location, CPGs at subtelomeric regions showed stronger hypomethylation compared to non-subtelomeric sites. This is consistent and moderate across patients with p-value of 0.005. So aggressive disease genes aren't just losing methylation randomly, they preferentially affected near chromosome ends. That was where they actually have their effects mostly. This schematics is just showing chromosome, looking at the ends called the telomeres, the area with gray shade at the subtelomeric region, and this indentation are the centromeres. And the blue ovals are the CPGs, which were randomly distributed across, just to give us a background of my next slides. So we mapped genome-wide value of this methylation. Significant CPGs were seen across multiple regions, but a striking enrichment at the subtelomeric level was evident on the short and long arms of chromosome 10. and long arms of chromosome 12 and 13. This builds on the previous discussion where we showed that aggressive disease genes display preferential hypomethylation at subtelomeres, suggesting that these regions may actually act as a focal hotspots in glioma evolution. Copy number analysis reinforces the methylation signal. Chromosome 10 loss increases stepwise with grade escalation, particularly on short arm of chromosome 10, and progressors show over 5-fold odds of loss compared to non-progressors. Taken together, this data highlights chromosome 10 as a central axis of glioma evolution and malignant transformation. To summarize this arm, in our longitudinal HG set, global DNA hypomethylation switches on with progression, not recurrence. And in the 3 time point cases, it deepens stepwise. The genomic cluster at the subtelomeres with hotspot on 10p and 10q, and also on 12q and 13q. Notably, although all chromosome 10 loss is the hallmark of glioblastoma, in our LGG cohort, we see earlier focal or segmental loss on chromosome 10, and this tracked with great escalation and a more aggressive course. We then go to the other aspect of my project. We ask, can tumor microenvironment help distinguish recurrence from progression? To test this, we profiled matched LGG pairs using microarrays, MIBI imaging, and SPART package. This pipeline was designed to map immune and stroma features against clinical trajectories. For each patient, we selected paired initial and subsequent resections. And for each of these resections, we have 2 contrasting regions that we chose. One with low cellularity or atypia, CD45 and K67, and the other with high levels of these features. This ensured that both low and high activity tumor areas were represented on our TMAs. Each patient therefore contributed 4 cores, 2 from initial and 2 from subsequent. And while we started with about 29 matched matched samples for 29 patients for this TMA generation, only 21 patients were eventually included for our longitudinal MIBI profiling due to tissue loss, and some of them have low cell counts. Here is the MIBI workflow. Antibody staining with sensitive markers that we used. Next was imaging and segmentation. In total, we imaged 458 fields of view. The cell types were then identified by combining unsupervised clustering with manual annotation, followed by supervised classification using the SGBoost machine learning model. SPART package in R then enables spatial analysis of cell densities and colocalization. Here is the representative spatial map, cell map with major neural, immune, and tumor compartments, which were defined by their corresponding markers. This multiplex approach enables direct comparison of tissue architecture between recurrence and progression. We observed that oligodendrocytes and neurons, which are termed normal-like compartments, are enriched at the tumor leading edge. Previous cross-sectional studies show that preservation of these normal-like programs associates with less aggressive biology, and their loss was linked to poor prognosis. This was established in glioblastoma, but here now we're establishing this one in low-grade glioma. Extending this insight, our matched longitudinal spatial cohort provides direct time-resolved evidence. Oligodendrocytes and neurons were significantly higher in recurrence compared to progression, especially at the leading edge. This demonstrates that preserved neuroglia compartment marks recurrence, whereas their depletion defines progression and correlates with worse prognosis. Looking at the CD8 T cells, density rose in both recurrence and progression, but only progression show a consistent significant expansion across all the patients. Notably, the expanded fraction was enriched for exhausted CD8 cells, which suggests that there was an activation, but this activation was not effective. It does not give effective anti-tumor response. When we map spatial proximity, CD8 cell in progression showed greater colocalization with tumor cells, both the IDH mutant and the IDH wild type. This suggests immune recognition is taking place, but tumor clearing is failing. Together, this points to immune dysfunction as a hallmark of progression, going beyond the simple immune exclusion that we all know. We next look at the myeloid arm. Macrophages trended up in progression, although we didn't see a clear subtype shift between M1 and M2. In contrast, dendritic cells were significantly reduced in progression, suggesting a weakened antigen presentation capacity, which could further undermine the TCL priming. Bringing this together, progression was marked by reduction in oligodendrocytes and neurons, especially at leading edge, in addition to expansion of CD8 T cells and exhausted CD8 T cells. Also, reduction in dendritic cell and upward trend in macrophages. All these features point to ineffective or a suppressed immunity. In summary, LGG progression is defined by loss of protective normal-like glia neural context, together with immune dysfunction, a hallmark that goes beyond the simple known theory of immune exclusion. This is just a pictorial representation of the outcomes of my finding. You could see here the recurrence, which should have a better course compared to the progression that have this aggressive course. And that in progression, there is reduced normal-like oligo and neuron, and also reduced dendritic cells. Also, we saw an upward trend of macrophages, increase in CD8 T cells, but the CD8 T cells subset were mostly the ineffective exhausted CD8 T cells. We saw a global hypomethylation that trended well with progression. And lastly, we saw focal segmented chromosome 10 loss, which may bother the question, do whole chromosome 10 loss we see in glioblastoma occur earlier, or is this another entity that we need to look further into? Future directions, larger cohorts. In my specimen, I was only able to investigate 5 patients that had at least 3 time point surgeries. So we are looking at higher cohorts of sample, and to be able to look at this trend in chromosome 10 and the global hypomethylation. Secondly, we will do independent validation with whole genome sequencing or SNP array plus fish spot checks in tissue to confirm what we've seen through methylation. And thirdly, we'll repeat this maybe at match time point to test whether immune shifts with or tracks with chromosome 10 loss. If this pattern holds across subsets, the patient impact will be meaningful with a simple, reasonable risk stratification combining chromosome 10 loss, low methylation, and immune context. Exploration of blood cell-free DNA or for less invasive monitoring may then be considered if this goes through following validation. This earlier risk signal will support more personalized management and follow-up of patients. I want to thank everyone who has contributed to my PhD journey. Like Sarah said earlier on, when I came into the lab 3 years ago, I was clueless, and most of what I worked with were things that I've never seen where I was coming from. But through patients and my friends in the lab and all the lab members, they guided me through, and I was able to achieve significant milestones. So I thank everyone in the lab, my supervisors and my beautiful lab members. Thank you very much for the support all this while. Thank you. The education department was very helpful because I think I've only been to University of Melbourne just once. But most of what I've done were through the education team, and they guided me through, very supportive, especially Surangali. The brain registry That was where my data was collated. And of course, Royal Melbourne Hospital, where I got all the samples I used for this study. My supervisor is here, Michael and Sam. They were very helpful, patient with me. Despite their busy schedule in pathology, they guided me through. The Gibbs lab ethics, oh my God, they've been very, very. I've been on application for ethics for some time now, and it has been giving me a tough time. But they stepped in, and they actually did the work. So kudos to the ethics team. For the maybe Claire and Michael, very instrumental. This special method was, my first time was for exposure, and they guided me through, patiently. So they were really helpful and with other things. Of course, the Brain Cancer Center, Sam and Linda, thank you very much. Through them, I got the Greg Simon top-up scholarship, which has been very helpful to my financial upwell. Thank you very much. Royal Prince Alfred Hospital, Lavinia and her team, very supportive, and Kaz and her team from Ontario Medical Research Institute in New South Wales, very helpful. Of course, my PhD committee. Sarah said I had some challenges with computational techniques when I started, because I've been practicing pathology. I don't know what is R, what is Python. And so when I started working on my PhD and I was having code, I was having a headache. But my PhD committee was like, you're a pathologist, you need to do something that can relate with your background. And that was where a change was made, and very supportive, open to hear what I have to say. Very, I really thank you for all you've done. Prof, thank you, and your other members. The division coordinator, Sabine, you, and other team members, thank you for all the timely emails, even though I respond to them very late, but thank you for all those emails. The entire POD has been my family, working in the same space environment over time. They've been very supportive, and I thank you so much for what you've done. Most time when they put me on let's restock, I even forget. They do it for me. I was like, wow, thank you for all you've done for me. WEHI histology, Ellen and team, very helpful. The TM generation was very difficult because we had to take samples from archives, and they were very difficult to construct TMAs. But Ellen took her time and helped me through. Thank you so much. To the Greg Simons family for the support you gave me. For the WEHI IPSI scholarship, wow, that was wonderful. When I got to WEHI here, I didn't realize that how fortunate I have been to be one of those people, one of the beneficiaries of IPSI. It's a very highly competitive program. Thank you for counting me worthy. And lastly, I traveled last year to do a step down of my PhD work in my home institution. The Eddie Moffat Travel Scholarship was very handy in doing this. Thank you so much. Of course, I can't forget to share this beautiful moment. This is my wife, my support in Australia, and here we were pregnant with our babies. And then, we take this picture. These are our parents, aged, but, very agile and strong, and we communicate all the time. When I traveled for my step-down PhD meeting in December, I had to visit my son. When I was coming to Australia, I was taller than him, but when I went back home, he was taller than me. I can't imagine how tall he will be now. This was when my wife was in the hospital for our baby. Her name is Rere, and this is Rere in the picture here. She's here with us today, and of course, I attended a number of conferences and workshops. This was in Accra in Sydney, and this I also took. I went out with Jorgen and some other team members. This is we took this picture, and this is London, my first conference in London with my bosses and my colleagues, and then I had to snap this as well. This day, I was tired in the lab. I haven't started work, and my eyes were just too dimmy. I just said, let me snap myself in case it becomes handy in the future. And of course, it's not just all about PhD. I have some extra PhD activities. And you can see here, this is my church where I was speaking on this day to the youth. It was a youth program, and it was a great program. So thank you all for everything you've done. I really appreciate your time.
Well done, Oluwaseun, on a beautiful completion seminar. We've got time for questions. All right, you can run around.
You ready for this, Oluwaseun?
Yeah, try as much.
In progression, you see increased CD8, which you found to be exhausted. How does the increased density compare to a tumour, to a typical hot tumour such as melanoma? I just came up with that question on my own.
So in progression, we have this CD8 T cell that has higher density compared to recurrence. And then when we probe further, because normally CD8 they are anti-tumor, they're expected to kill the tumor cells. But we found that the subset of those T cells are actually exhausted T cells. They've been primed, they've been called to action, but they fail to have that anti-tumor activity. So this we saw that was more in progression, meaning that in progression, there's attempt at limiting the progression, but the attempt actually failed, which could have been what's responsible for why the tumor keep growing and then keep transforming to a different grade. As to how melanoma, right? I've not really examined melanoma, but I think it's something that I can look into. Thank you.
Congrats, Oluwaseun.
Thank you.
I was just wondering whether you could comment a bit more on the hypermethylated CpG hotspots that you saw. So obviously there were the ones that were close to the telomeres, but then there were also all the other ones. Were there some that might be close to tumor suppressor genes or proto-oncogenes, or did they seem quite random?
Okay. Thank you very much. We examine those CpGs and to see, because this is an epigenetic evaluation, most of the CpG were at promoter levels, which means that when they are hypomethylated, they tend to cause increased gene expression. When they are hypermethylated, they cause decreased gene expression. So all the 23 genes that we're able to collate as aggressive disease gene subsets were at the promoters, so which, of course, the other data we show in the thesis, but for the sake of this presentation, I couldn't add all those data, but they were at the promoter region, and so they were very important. Then we have enhancers and some other regions, but these genes were the promoter regions, not just scattered anywhere, even though they were at the subtelomeric, but they were the promoter region of the subtelomeric, which is between the less than 200 kilobytes from the chromosomal end, and then they were all promoters. We have a heat map regarding that, and that shows those genes, whether they are enhancer, promoter, or any other areas of the gene. Thank you.
Thanks, Oluwaseun. Beautiful talk, and really nice to see the work coming together like this. My question is related to RAF, but I'm going to take it a bit of a different angle. In the hypomethylated genes that you've identified so that they're now upregulated, is there any correlation with the role of those genes in changing the microenvironment where you see those change in macrophages, T cells? Can you link, the two aspects,
Thank
the
you.
MIB and the hypomethylation work?
Thank you, Ma. So, like I earlier mentioned, when we noted this shift in hypermethylation, one of the things that I want to take further, because it would be good to investigate this, not just with with 2 time points, initial diagnosis and subsequent, but where we have more, time points, at least 3 time points, to be able, we are already taking stock of samples from other institutions in which some of them have 3 time points, 4 time points. So when we have these tumors, so we then investigate, are there changes in the immune microenvironment with these methylation changes, so we can investigate beyond just the 2 time point and extrapolate and be able to come to a more evident conclusion. Thank you.
Hello, great work. Congrats on your PhD. I'm just asking a question about your normal-like oligocyte. Did I say that cell type right?
Normal-like, yeah.
Yeah, cell type at the time of initial sampling before recurrence, and that is reduced in those that progress. Is that correct? What is your speculation on that mechanism of progression? Do you think it is linked to some of your hypomethylation allowing for a greater transformation, or do you think this is just a bystander effect?
Okay. The normal-like, as defined in literature, are the non-tumor component, and mostly the glial cells, the astrocytes, the oligodendrocytes, the neurons that form the environment where the tumor grows. So from literature, for GBM, it's been established that when you have this normal-like, mostly, especially at tumor leading edge is where the tumor grows into the normal brain area. When you have a handful of them, they tend to limit how tumor invade and even progression. However, when you have them very few or scanty, not quantified, there is no cutoff for now. But when they are few, then it tends to associate with tumor progression. And that was in GBM, and that was what we observed in the LGG, that at the leading edge, we saw more neurons and more glial cells in recurrence. And recurrence has this indolent, less aggressive course. But we didn't see much of this in progression, which has a more aggressive clinical course. So which also aligns with what has been established in glioblastoma, that this normal-like is linked to whether a tumor will progress or have a poor prognosis or not.
Thank you.
Do I answer the question? I'm not sure.
Oh, sorry, I think so. So are they like a barrier or something?
What's it?
Are they a barrier? Or?
Barrier, it's not really a physical barrier. It's more or less like tumor density versus normal cell density.
Okay.
Yeah.
Thank you.
Nice.
And just to follow up on that, was there a correlation with the normal-like cells and the gross total resection?
Normal-like cell?
Yeah, could it have correlated with a gross total resection in the recurrent samples?
Yeah, they could. It's possible. Yeah.
Questions? This is our opportunity to grill Oluwaseun on his PhD. All right. Jürgen.
Yes, please.
Great talk, by the way. You focus on the in a patient-matched fashion, the recurrent versus the progressing samples, and showed the changes. What about comparing, so you showed the slide looking at the different clinical courses of different patients, how could they're very different. So going to the initial samples, the ones that progressed versus the ones that recurred, could you sort of predict any, or are there any sort of methylation changes or microenvironmental changes that were different between those patients?
That will recall, and those who will not recall?
The ones that recurred versus the ones that progressed. So the ones that recurred in the same grade and the ones that recurred as a higher grade, were there any methylation differences or microenvironmental differences that would allow you to predict those in an earlier time point?
Yeah. So those that recall, that is, those that occur at the same grade level, initial grade 2, later grade 2, and those that transform or progress, initial grade 2 and perhaps subsequent grade 3 or grade 4. Yes, so we saw that globally, those that progress has hypomethylation.
All right, so are those in the initial samples, not in the recurrence?
No, the hypomethylation, it's a matched sample. So now, how can we tell whether at initial stage, how do we tell which will progress or not? The map I showed was the initial of progression and the initial of, I showed the map of initial progression and also initial recurrence. So at initial stage, first sample received, you could tell when you look at the methylation profiling, those with high methylation may likely recur, and those with low methylation may likely progress. The question there that we've not been able to answer is that there's not yet been a cutoff, the value cutoff that you can use. Currently, what they use is the mean, the methylation mean for each cohort. But I think we should be able to get to a place where we'll be able to, look at a defining cutoff to use so that we can differentiate them, with all confidence at the initial presentation.
Yeah, got it. No, thank you. That's very clear.
Right. Quick last question. Yeah.
Hi, Oluwaseun. Beautiful work. Can I ask you a technology question?
Yeah, please.
So you did a lot of work and platform development on the MIBI, and now the MIBI has gone bust. But I mean, there's so much spatial proteomics work that you've done. How easily transferable would that information be to like other antibody-based or newer spatial proteomics platforms, given your panel and segmentation work that you've done as a baseline?
Okay.
Yeah.
Maybe it's one of those spatial proteomic technique which we adopt because of some of the advantages it has, multiple panels, and then can be used on previous FFP sample. So those are things that determine why we use MIBI. Yes, this work can actually be repeated on other spatial techniques, using the antibodies that we use in this MIBI. So I don't see it not reproducible with other techniques. Although I haven't tried that, but I think it's a beautiful thing to look into. And you are the technique person, so I think we can actually, use it on, because like you said, maybe it's quite expensive and it requires maybe a lot of work and knowledge, but there may be some other special technique that may not require so much work that can make maybe a little easier. But because we want to demonstrate the quality of our work, that was why we went for MIBI, but it can be reproduced with other systems.
Great, thanks.
Great. Well, congratulations, Oluwaseun, on completing your PhD completion seminar.
Thank you, everyone.
Well done.